Good evening, everyone. Just confirm me that everything is perfect. Am I audible? Whether the PPT is visible or not, just make sure everything. Everything is okay, right? Just wait for 1 minute and we'll start our class for today. Okay, good. Okay, fine. Okay. So we have completed three important chapters. That is non-chordata, chordata, and cell biology. Okay. And for today, I'm going to cover the genetics. And the first topic that we are going to discuss today is Mendel's law of inheritance. But before that, I want to show you the previous question papers with respect to this cell biology. See, what are all the things we have discussed in the MCQ series, okay, regarding the cell biology. All the questions have reflected in the question paper, in the previous year question paper. Just answer for this question. Identify the function of the nucleolus. Identify the functions of nucleolus. We have discussed everything detailedly. Just give me 2 seconds. I'll do that soon. Okay, what is the function of nucleolus? Very good. What is that? Biosynthesis of ribosomal units. Okay, we have discussed this question. Okay, now, which of the following are the mitochondrial functions? Electron transport coupled with ATP production during glycolysis, fatty acid breakdown, production of reduced coenzymes. See, just remember what is a function of mitochondria. If you know that, you can easily come up into an answer. Very good. And ATP production during glycolysis is taking place in which area? ATP production during glycolysis is taking place in very good. And also you have to understand how many number of ATP is produced in glycolysis. What is the gross ATP produced in case of glycolysis? And also you should know that what is the net ATP produced in glycolysis. Okay. And after that, you also have to understand how many number of ATP produced in case of mitochondria. Okay. What is the gross and what is the net value? This is your duty and you have to know that. You have to read that. And overall cellular respiration, if they ask the question like this means how many number of ATP will be produced in cellular respiration means then you have to say that 38 ATP molecules. That too gross ATP will be 38. Okay, in case of a net ATP, then it will be 30 6. Okay, there will be the difference between gross and net. Gross means total. Gross means total. Total ATP molecules produced in the cellular respiration will be 38 ATP molecules. And how many will be get as a net ATP means, then you have to say 36 ATP molecules. Likewise, you have to read individually. For example, glycolysis, mitochondria, like that. Sorry, glycolysis and then the Krebs cycle. Okay. Which of the following is true with respect to smooth endoplasmic reticulum? Main functions of smooth endoplasmic reticulum. See how the questions are very simple. See, so you have to you can easily get you can easily score the marks in your examination. If you know the basic knowledge, it is sufficient to answer these kinds of questions. What is meant by hepatocytes? What is meant by hepatocytes? What is meant by hepatocytes? Very good. The cells which is present in the liver, which is called as hepatocytes. And what is the main functions of liver? What is the main function of liver? What is the main function of liver? What is the main function of liver? Very good. Detoxification. And which organelle is responsible for detoxification? Which organelle is responsible for detoxification? Smooth endoplasmic reticulum. Okay. Yes. Smooth endoplasmic reticulum is responsible for detoxification majorly. That is why smooth endoplasmic reticulum is majorly present in liver. Okay. Fatty acid synthesis. Lipid synthesis is the main function performed by this SER, smooth endoplasmic reticulum. Okay, and this is regarding to the rough endoplasmic reticulum. And as usual, it is abundant in the liver cells. Okay, and synthesis of phospholipid. Again, it will be the lipid. This is how you have to read. That is why I'm showing the question paper. Okay, and DNA polymerases add. We know that the DNA polymerase is involved in the DNA replication. And see, they are adding the nucleotides. That will be the predominant function performed by this DNA polymerase. And in which direction? This is what a question. Always, the daughter strand synthesis will be held at which direction? See how the questions are very simple. Obviously, that will be the 5' to 3' direction, and DNA polymerase is somewhat related to the DNA, that is deoxyribonucleic acid. And the F0, F1 particles in mitochondria are located in the same question I have taken for our MCQ series. The F0, F1 particles in mitochondria are located. Yeah, that is obviously the inner side of the inner mitochondrial membrane. Okay, and it is also called as the ATP synthase. And which of the following is not concerned with initiation of transcription in eukaryotes? eukaryotic genes. Which of the following is not concerned with initiation of transcription in eukaryotic genes? See, what are all the, see, don't think too much. Just see the first option. This is DNA polymerase. Do you think this DNA polymerase is involved in the transcription? Just think it. And RNA polymerase. Do you think this enzyme involved in the transcription? And CTF, TF. This is somewhat related to the transcription factors. Obviously, it has to be associated with the transcription. And RNA polymerase is generally what is transcription? It is going to synthesize an RNA strand by using DNA as a template. Okay, so for that, RNA polymerase is definitely important. And this DNA polymerase is somewhat related to the DNA replication. So obviously, this DNA polymerase binding is not concerned with the process of transcription. Okay. Answer for this question: promoter, splicing, 5' capping, start codon. Each and every word we have discussed in our class. What is a start codon? And what is mean by 5' capping, splicing, promoter? Who said? Okay. 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 Start codon is AUG. What is the name of an amino acid which is code for this AUG? What is the name of an amino acid which is going to code for this AUG? Very good. Methionine. Okay. So if you know this thing, you can easily come up into one answer as B. And 5' capping. 5' capping is somewhat related to the 7-methyl guanosine. Methyl group is going to add the guanosine at the 7th position during the process of capping. And what is the splicing? It is a primary transcript, which means what? Which is going to remove all the introns and going to join all the exons. Okay. And what is a promoter? Again, it will be associated with the Tata box during the transcription. Okay. This is how the question will be. Okay, you don't want to put an extra effort to read all those concepts and all. If you know the basic concepts, you can easily answer these questions. And once again, which one of the following is related to the cytoskeleton? Okay, what is gibberellin and auxin? They are somewhat related to the plant hormones. Okay, and what is glomerulus? It is related to the kidney. And microfilaments and microtubules will be the cytoskeleton. Okay, microtubules, microfilaments, and intermediary filaments. These three are related to the cytoskeleton. And who is going to produce? Tubulin is the motor protein of which cytoskeleton? That will be the microtubules. Okay. That's all about the questions. And yeah, one more question is here. Purification of maturation promoting factor, that is MPF, revealed that it is a composed of purification of maturation promoting factor revealed that it is composed of cyclin and cyclin dependent protein kinase, cytochromes and nucleosomes, cDNA, that is complementary DNAs, ATP and GTP molecules. Obviously, that will be the cyclin and cyclin dependent protein kinase. Okay. Okay. I think you have got an idea somewhat related how the question has been asked in your examination. This is the standard of your question. Okay. So you have to thorough with your basic information. So for today, I'm going to discuss genetics. Okay, that too particularly the Mendel's law of inheritance. How many law has been proposed by Mendel? 3 law. That is law of dominance, a law of segregation, and law of independent assortment. Okay, I hope you all know these things. And what is the ratio of phenotypes resulting from a monohybrid cross between the heterozygous individuals according to the Mendel's law? Ratio of the phenotype, particularly the monohybrid cross between two heterozygous individual. Heterozygous means what? Like this. Okay. What will be the monohybrid cross? We have to take only one character, okay, with the two variations. We have to take height as a character with the two variations, whether it is a tall or dwarf. Okay, this is a pure plant breeding, pure tall and pure dwarf. And what will be the gamete will be present here? One tall and one dwarf. So the F1 generation will be like this. Okay, everything will be like this, heterozygous. So in the F2 generation, we are going to cross using the Punnett square like this. Okay, see, this will be the tall, pure tall, that is homozygous, and this will be the also tall, and this is also tall, and this is the dwarf. Okay, what is the question asking here is phenotypes. What is the morphology, whether it is tall or dwarf? This is the question. See, 3 tall, that is 3, and 1 is dwarf. And what will be the genotypic ratio of the monohybrid cross? What will be the genotypic ratio of the monohybrid cross? What will be the genotypic ratio of the monohybrid cross? How that is 1:2? Because a pure tall, that is homozygous tall, will be the only one. Okay, only one combination. But here it is also tall, but it is heterozygous. That is why we are having this two. In a genetically, that is with respect to these genes, it has a one dominant character and one recessive character. Genetically, the dwarf is also present, but it has been masked by the dominant character. Okay. And also the pure dwarf will be only one. Okay. This is what a difference between a genotype and a phenotype. Now, which of the following represents the law of independent assortment? Law of independent assortment. And my question is, in which cross, okay, whether it will be the monohybrid or dihybrid or trihybrid, in which cross Mendel gives this particular law? In which law, in which cross, which type of cross Mendel proposed this law of independent assortment? Very good. That will be the dihybrid cross. Okay. So, see, you have to understand the difference between these two. Alleles for a different trait and alleles of a for a same trait. Okay. But as it is a dihybrid cross, obviously the characters have been segregated independently from different characters. Okay. So, this law of independent assortment have been brought by the Mendel from the dihybrid cross, and you have to make a note of this particular law is mainly it is from the different traits, okay, not from the same trait. Okay. Mendel's law of independent assortment states that alleles for a different trait segregate independently during the formation of a gametes. This means that the inheritance of one trait is not dependent on the inheritance of the another trait. Whether the other trait is going to inherit or not, that is immaterial for the particular trait to inherit. Okay, this is what law of independent assortment. Now, in a dihybrid cross between two heterozygous individuals. Okay, dihybrid cross between a two heterozygous individual. What is the phenotypic ratio according to the Mendel's law of independent assortment? Whatever the question may be, okay, you can think how the question may be, that is immaterial. Okay, you have to read this statement whether it is a dihybrid or monohybrid, and whether you have to see whether they are asking phenotypic ratio or genotypic ratio. So obviously the phenotypic ratio will be the 9 is to 3 is to 3 is to 1. Okay, and my question is, and in which whether it is 9 or 3 or 3 or 1. Okay, you have to see this 3 refers to which number is refers to phenotype with dominant allele for one trait and recessive allele for the other. What I'm asking is heterozygous individual. Dominant allele for one trait and recessive allele for the other. That will be 3. See, this 9 is refers to as the dominant for both the trait, which means what? Like this. Okay. A, B, B. This is what 9 refers to. And this 1 is for recessive for both the trait. Like this. Okay. And this 3 is for like this. Okay. And this 3 is for like this. Okay. This is like this. Okay, this is what this 3 and 3 are heterozygous. One is dominant and one is recessive. One character is dominant and other character is recessive. So you have to aware of all the statements, how they are asking and what they are asking. And same thing in a dihybrid cross between the two individual heterozygous for linked genes. For two linked genes, what phenotypic ratio is expected according to Mendel's law of linkage? Here they are asking in connection with the linkage. In a dihybrid cross between two, that is heterozygous individual. And they are saying that these are the linked genes. What will be the phenotypic ratio? How it will be? See? Can you explain? See, that will be 1:1:1 because all offsprings have the same phenotype because they are linked. Okay, all these are linked. So obviously that will be reflected. What are all the parental combinations? Same will be reflected even though in the offsprings. Okay, so this is what a difference. In linkage, which process is absent? In linkage, which process is absent? Why it has been reflected as like in the parental combination? Why the offspring is just looked exactly as the parents? Is it so? Is it an assortment? Okay. Which process is absent here? Very good. What is the name of the process? That is crossing over. The recombinant genes will not be seen because crossing over is absent here. Why crossing over is absent here? Because all the genes are linked here. As it is linked, what are all the characters seen in the parental combination? Same will be reflected in the offsprings also. Okay. Which of the following situation would most likely result in the recombination frequency of 15% between the two genes? Anyhow, I'm going to explain the linkage and crossing over and recombination frequency, everything and all. So for that, just tell me, just give a try. Here they are saying which situations is most likely result in the recombination frequency of 15%. So here you have to see the crossing over is happened here. And when the crossing over will happen? Crossing over will happen between the two different chromosomes. Okay. If it is very close, what will be the name of gene? If it will be the far apart, what will be the name of genes? If it is located on the different chromosome, whether the crossing over will occur or not. Okay, so these are all the different situation. And what will be the answer? C. Always crossing over will occur whenever the genes are present in the different chromosomes. See, this is what a chromosome from a mother. This is what a chromosome from the father. So if they have to produce a progeny, definitely the crossing over has to happen between the two non-sister chromatids, that is from different chromosomes. Only then by mixture of the recombinants will be happen here. This is why we are calling them as the recombinants. Okay, see, let me explain you in detail, right? This is one chromosome and this is the other chromosome. Now, this is the area from one chromosome and this is the area from the other chromosome has to cross over. What are all the genes present in this chromosome will go here and what are all the genes present here will come here. As a result, see how the how the genes are segregating here. One gene like this, see, because this is the combination where no recombinants will be present here. But in here, we have a recombinants. So as a result, see 50% of the recombination has been happened whenever the genes will be present in the different chromosomes, because see in totally we are getting 100%. Okay, and 2 will be recombinants. Remaining 2 will be half of the chromosome will be a recombinant, half of the chromosome will be the parental combination. So we can get the recombination frequency will be 50% here. Okay, this is how you have to understand. This is how you have to calculate the recombinant frequency, whether it will be the 50% or whether it will be the 20% like this. Okay, clear? Everybody clear? Before we'll solving all these questions, I'll explain the linkage and crossing over, and after that we'll solve these. Okay. Okay. Let me ask you a simple questions before that. Linkage and crossing over will be discussed detailedly for tomorrow. Okay. And do we have any questions from that? Okay. There is not much question from the Mendel's law of inheritance. You have to focus on the what is monohybrid, dihybrid, and dihybrid, and you have to focus on the what is the phenotypic ratio and genotypic ratio, and what is law of dominance and segregation and law of independent assortment. This is what related to the Mendel's law of inheritance. Okay. For tomorrow, just read and come the linkage and crossing over. This is what a test for tomorrow. Okay. Any other clarification you want to know? That's all for today. Do you have any doubts? Or everybody clear? Okay, this is the test for today. Tomorrow, linkage and crossing over, you have to finish and pump continuously. There will be no gap in the middle. Okay, we are going to cover the entire syllabus through these MCQs. Okay, don't worry. Okay. Is there any other clarification you want to know? Okay, fine. Then we'll meet tomorrow at 8:30 p.m. sharp. Linkage and crossing over will be the test for tomorrow. Okay, thank you. Thank you, everyone.